Hi, did you know that your body builds DNA and RNA from scratch, one atom at a time? That's the magic of nucleotide metabolism. I am Dr. Juliet. Welcome to Julsummit Biochemistry Hub, your go-to place for mastering biochemistry, one concept at a time. Today, we shall be diving into nucleotide metabolism, specifically the de novo synthesis of ribonucleotide pyrimidines. The synthesis of pyrimidines occurs in 6 enzymatic steps in the liver. Some of the steps, step 1 and step 4, occurs in the mitochondria of the liver cells, while the rest steps 2, 3, 5, and 6 occurs in the cytosol or cytoplasm of the liver cells. We must remember that pyrimidine rings are synthesized free before they are attached to the ribose sugar, which we will see in step 5. The first step in pyrimidine synthesis is the reaction between glutamine, bicarbonate, water, and 2 molecules of ATP. Glutamine donates an amino group. The bicarbonate ion donates a CO2 group, and one of the molecules of ATP donates a phosphate group. So that leaving that reaction, we have glutamate, which is glutamine minus an amino group, and we have 2 molecules of ADP and 1 molecule of phosphate. One of it has been left behind, and this compound is called carbamoyl phosphate. This reaction, as step 1, is a very important step and is a regulatory step for this entire pathway in animals. The enzyme carbamoyl phosphate synthetase 2 is a regulatory point of the synthesis in animals, while for bacteria it is regulated as step 2. The next step in this synthesis from scratch will be the conversion of carbamoyl phosphate to carbamoyl aspartate. What actually is happening here? Aspartate comes in to combine with carbamoyl phosphate at this point of attachment to the phosphate group. So aspartate, the whole of the amino acid, takes the place of this phosphate, and phosphate leaves in this reaction, catalyzed by the enzyme aspartate transcarbamylase. And so we can see here in the product of that reaction, carbamoyl phosphate, we can see that phosphate has been displaced and replaced with aspartate amino acids. So COOH, CH2CH, COO, and H is aspartate amino acid to give the compound carbamoyl aspartate. Now proceeding forward to step 3, what happens here is now a closure of the ring. Consider the ring is the open here. And so here water is removed by the enzyme dihydroorotate. The water molecule that is being removed is just this OH is removed and 1 hydrogen is removed from here. So we now have CO left and NH, and you can see the closure of the ring. Water is removed. As step 4, which is catalyzed by the enzyme dihydroorotate dehydrogenase, quinone comes in as a releasing agent and removes 2 hydrogen atoms from dihydroorotate to give us orotate. Now where did this hydrogen come from? 1 hydrogen is removed from here and 1 from here. So we now have a double bond in between these 2 carbons. It's no longer CH2, it's now CH, no longer CHCO minus, it's just now CCOO minus. So we can see a double bond here. And then at step 5, this is the free pyrimidine ring. It's been synthesized as orotate, and now phospholipase is added to it at this step, catalyzed by the enzyme orotate phosphoribosyltransferase transferase, so that phospholipase pyrophosphate comes in to react with this, and then pyrophosphate is released. So what we have here is a phosphorylated sugar added to this entire ring at nitrogen 1. That's what we have here now. So we now have, it's no longer orotate, it's now orotidine monophosphate. Remember, when the free bases, pyridine base, add to join to sugar. They end in idine from our previous class. So it's no longer orotate, it's now orotidine monophosphate. And that's what we have here. Sugar, phosphate, and nucleobase. So this is a nucleotide. And then the last step, we need to lose this this CO2 group. And so it is lost here at step 6, catalyzed by the enzyme orotidine monophosphate decarboxylase here, or it's also called UMP synthase. Removes CO2. This CO2 is removed and replaced with hydrogen at this step. So we have our first parent pyrimidine ring, uridine monophosphate, which is what we have here. So we can see a 6-step reaction gives birth to uridine monophosphate, our first parent pyrimidine nucleotide. And at this point, step 1 and step 4 occurs in the mitochondria of the liver cells, while the rest steps take place in the cytosol. But we also remember this is not the only pyrimidine that we have. We also have cytidine monophosphate, we have thymidine monophosphate. So from this step, we can get the other two from uridine monophosphate. The first thing that happens is that monophosphate is converted to uridine diphosphate by the enzyme UMP kinase as uridine monophosphate kinase, which gives a phosphate group to it, one of these, and then the next step to give uridine diphosphate. And in the next step, nucleoside diphosphate kinase adds another phosphate group to uridine diphosphate to give us uridine triphosphate, which we will have here. What's the difference? In uridine monophosphate, we have just one phosphate group attached to the sugar, but now in uridine diphosphate, we have 3 phosphate groups attached to the ribose. And then it is at this point that we can synthesize cytidine nucleotide from uridine nucleotide. How do we do that? Glutamine comes in with the help of water and a molecule of ATP to donate its amino group to UTP at carbon 4. So this oxygen atom is replaced with an amino group from glutamine and it leaves as glutamate with the help of hydrolysis of a molecule of ATP to ADP and phosphate group. So we have this phosphate. So we have this as a final product, cytidine triphosphate, which naturally gives off its phosphate groups to return to cytidine monophosphate. And this enzyme is catalyzed by cytidine triphosphate synthetase enzyme. Now, I trust that you appreciate what has happened here today. In the next class, we'll be looking at the novel synthesis of ribonucleotide purines, and then we'll be comparing the step, which is more complex, to this particular step. If you found this helpful, kindly hit the like button, follow us on Facebook, YouTube, and TikTok for more enlightening lectures and biochemistry. Thank you for your time today. Until see you next time. Bye-bye for now.